My name's Sam, and today we're going to talk about something that we all love, food. We need it nutritionally for our body, and we love to eat it. But nobody really knows what happens after we eat our food. So let's go find out.
Our body breaks down the food we eat with gastric juices, and a major part of these juices is HCl. In this episode, we'll take you through how HCl is formed, what it does, how it's secreted, and what factors influence secretion. CO2 enters the parietal cell through the blood, which binds with H2O that produces H2CO3 or carbonic acid. The combination of CO2 and H2O to create carbonic acid is catalyzed by carbonic anhydrase. The carbonic acid then splits into an H plus proton and HCO3-. H plus is actively transported to the lumen in exchange for potassium through an H plus, K plus ATPase pump. HCO3- then leaves the parietal cell and enters back in the blood in exchange for a chloride ion on the basal side of the cell. The blood becomes more basic due to the higher concentration of HCO3-. Cl- then leaves the parietal cell down its electrochemical gradient through a luminal Cl- channel into the lumen. Cl- binds with H plus to create HCl. Now let's take a look at the functions of HCl. HCl activates the enzyme precursor pepsinogen to an active enzyme pepsin and provides an acid medium that is optimal for pepsin activity. HCl aids in the breakdown of connective tissue and muscle fibers, thereby reducing large food particles into smaller particles. HCl denatures protein, that is, it uncoils proteins from their tertiary structure, thus exposing more of the peptide bonds for enzymatic attack. Lastly, HCl, along with salivary lysozyme, kills most microorganisms ingested with the food, although some resistant ones may escape and continue to grow and multiply in the intestine.
So the first phase in gastric secretion is called the cephalic phase. This refers to the increase in HCl and pepsinogen in anticipation of food, so before food enters the stomach. This is governed by vagus nerves. Vagal stimulation leads to two things. It leads to an increase in acetylcholine, which leads to an increase in HCl and pepsinogen secretion. It also triggers G cells to release gastrin. Gastrin promotes histamine secretion, which leads to more HCl in the stomach. The second phase of gastric secretion is called the gastric phase. This occurs when food enters the stomach. Stimuli in the stomach can include distension, alcohol, or caffeine, but the biggest one is protein. So protein stimulates chemoreceptors. This activates intrinsic nerve plexuses, which leads to increased secretion by secretory cells. The last phase of gastric secretion is called the intestinal phase, which refers to the factors originating in the small intestine that influence secretion. The presence of the products of protein digestion in the duodenum stimulate more gastric secretion by stimulating increased gastrin.
So Sam talked about somatostatin. Somatostatin is released to reduce gastric secretion when gastric pH gets too low. Now there are other ways to inhibit gastric secretion. One of them is called emptying. Since protein is the main stimulus in the stomach for gastric secretion, when the protein is removed from the stomach and goes into the duodenum, gastric secretion is naturally reduced. Now the same factors that inhibit gastric motility also inhibit gastric secretion, so things like fat, high acid, or hypotonicity. The enterogastric reflex and enterogastrones also suppress gastric secretion when they reduce the excitability of gastric smooth muscle cells.
So after highlighting the process of how HCl secretion occurs, we thought it would be important to include a little bit of information on what factors actually control secretion. And as you can see, there are 4 of them. So specific to HCl secretion, acetylcholine, gastrin, and histamine all are stimulatory, that is, they increase secretion of HCl by promoting insertion of hydrogen and potassium ATPases into parietal cells' plasma membranes. Additionally, acetylcholine and gastrin also increase pepsinogen secretion by stimulating chief cells. Somatostatin, on the other hand, actually inhibits HCl secretion, as you can see by our awesomely animated little red arrow. Now let's take a closer look at these chemical messengers, starting with acetylcholine. Acetylcholine is a neurotransmitter released from the intrinsic nerve plexus in response to vagus stimulation. What does it stimulate? Acetylcholine stimulates parietal cells, chief cells, and HCl cells, all to secrete. And acetylcholine is also pretty chill in that it really doesn't inhibit anything when it comes to gastric secretion. Moving on, taking a look at gastrin. Gastrin is a hormone secreted by G cells in response to protein products in the stomach and acetylcholine, what we just talked about. Gastrin stimulates parietal cells and chief cells to promote secretion of the highly acidic gastric juice. Gastrin also indirectly promotes HCl secretion by stimulating HCl cells to release histamine. And gastrin, being a pretty busy neurotransmitter, also maintains secretory capabilities of the mucosa cells in the stomach and small intestine. Gastrin's also pretty chill in that it really doesn't inhibit anything when it comes to gastric secretion. Now let's take a look at histamine. So histamine is a paracrine hormone, throwback to last unit, released from HCl cells in response to both gastrin and acetylcholine. And now you can see why we chose this order of presentation. Histamine acts locally to increase the rate of HCl secretion. It also potentiates actions of both gastrin and acetylcholine. Finally, we have somatostatin. Somatostatin is also a paracrine hormone released from the D cells in response to HCl secretion. Somatostatin is actually an inhibitor. As you can see, here's our awesome red animation arrow again. It acts in a negative feedback to inhibit the secretion of parietal cells, G cells, and HCl cells. And what does somatostatin stimulate? Nothing. We also thought it would be important to highlight how the stomach actually protects itself from these highly acidic secretions so it doesn't end up like Jay Cutler on a Sunday against the Green Bay Packers. Go Pack. So the gastric mucosa is actually covered by a layer of mucus. Yum. This mucus serves as a protective coating that prevents the strong acid and prolytic enzymes from destroying the stomach. It also prevents self-digestion of the stomach since pepsin is inhibited with coming into contact with the mucus. And finally, it also neutralizes the HCl in the vicinity of the gastric lining. This gastric mucosal barrier actually secretes bicarbonate, and that's how it neutralizes this acid. The luminal membranes of these gastric mucosal cells are almost impermeable to hydrogen ions, so the hydrogen can't penetrate into the cells to cause damage. The acid actually can't diffuse between these cells into the submucosa, and finally, it's important to note that the stomach lining is replaced every 3 days.
Well guys, I'm Sam, I'm your host. Had a great time. We'll see you next time on Good Eats.